This is lecture 7, the cellular basis of inheritance, but in other words, meiosis or meiosis, whichever kind of helps you remember it better. So this comes from the Concepts of Biology OER textbook, chapter 7. So again, here are the learning objectives. That might sound kind of intimidating, but hopefully by the end of this section, you will be able to explain that variation from offspring is a potential evolutionary advantage, and that's resulting from sexual reproduction. Describe the behavior of chromosomes during meiosis. Describe cellular events during meiosis, and explain the differences between meiosis and mitosis. And then explain what are the mechanisms that make meiosis generate so much genetic variation as a result. So again, hopefully this will make sense by the end. So introduction. The ability to reproduce in kind is a basic characteristic of all living things. And so what does that even mean, in kind? So for example, in kind means that the offspring of any organism closely resembles its parents. So a hippo gives birth to a baby hippo, like Mudang. So it's not like a hippo is all of a sudden gonna have a baby that is a tree or is a flamingo. It's always gonna reproduce in kind, like it's gonna make more of its own kind. So a hippo is gonna have a baby hippo. When trees reproduce, they're gonna like give rise to the same kind of tree, and so on. So that's what the ability to reproduce in kind means. You are dividing and making another organism that's your own kind. So here's a little baby hippo. This was way back before Mudang, but good for her, you know, getting out there, getting big. So this happens because of sexual reproduction. So like I said, some organisms will reproduce by just dividing. So single-celled organisms will reproduce like that. But other more complex organisms usually divide by some other kind of sexual reproduction. And there are different kinds of reproductive strategies, but here we're going to focus on just like sexual reproduction. Yeah, if you look in the textbook, there are other kinds like fungi and certain kinds of plants. Like they have different kinds of reproductive strategies and cycles, but here we're going to kind of focus on one kind. So sexual reproduction is the production of specialized cells called gametes. So first you have to make these like specialized cells called gametes, and those cells will fuse with another gamete from each genetic parent to form a single unique cell. And then that fertilized cell will go through mitosis, go through a lot of different, mitotic cell divisions to develop into a full-grown organism. So this process that we're going to talk about today is different. So even though it's kind of like you're dividing a cell, it's a different purpose entirely. So we'll get into that in a couple slides. So comparing and contrasting those different reproductive strategies. So asexual reproduction is when offspring are genetically identical to the parent. So for example, like a bacteria. So one bacteria will just double up and then divide, and now you have two bacteria that are exactly the same. So in asexual reproduction like that, the offspring are genetically identical to the parent. It doesn't require another organism of the opposite sex. So one bacteria doesn't need to go find another bacteria of the opposite sex to reproduce, So it's kind of easier. You don't have to depend on like the other organism. Just one can divide. And then every individual is capable of reproduction. So it's not like only half of the population can reproduce because it depends on the other half. Every bacteria is able to divide on its own. So those, that is all asexual reproduction. And then we could compare it to sexual reproduction. And in that kind of reproduction strategy, the offspring are similar to the parent, but there's variation. So like we look similar to our parents, but we're not identical to our parents, and we're not identical to our siblings. And that's because of sexual reproduction. This strategy does require another member of the opposite sex. So it's not like one person can decide on their own. You have to find a way to get the gamete, of the other person. And then, so that means only half the individuals, have the anatomy to be able to reproduce. So there seem to kind of be more pros versus cons of asexual reproduction. You're not really limited by who can reproduce or needing another individual to reproduce. But that is kind of like a downside, that all of the organisms look exactly the same. So even though there might seem to be more pros and cons, why is sexual reproduction a thing? That's because of how much variation that is possible through sexual reproduction that it creates. And it's super important to the survival and reproduction of those offspring. So that's why sexual reproduction is even a thing. Like, even though it's not, it doesn't allow everybody to reproduce at any time, the fact that it is the way it is and produces so much differences and variation is a huge benefit to species that can do that, and that's kind of why it kept going. So we'll see how this process of meiosis produces some genetic diversity, and then all the different ways that there's like even more diversity. So like I said, there's different reproductive strategies, but we're just going to kind of focus on this one. So here is a life cycle in animals. So say we start with individuals. And so in each individual, they will have some cells undergo this other process called meiosis or meiosis, if that helps you remember it, separate than mitosis. So individuals will have some cells that undergo meiosis, and so this makes specialized reproductive cells, like sperm or egg cells. And so then these specialized reproductive cells will fuse together and fertilize. And so through fertilization, it makes one cell called a zygote. And the zygote is what will end up, dividing more and becoming, like a fetus, and then a baby, and then end up developing into a full-grown human. So cell division and mitosis is what kind of gets you from that one cell stage dividing into becoming a more and more complex multicellular individual. You're making more and more of the exact same kind of cells, and then you can, form a full-grown person. Meiosis is different because it's only making these very special kind of cells, sperm or egg cells, that will end up being part of this half of the life cycle, and then the rest is kind of dominated by this part. So that's how it all comes together. Like, they are kind of both making more cells, but they're very different in how and what's the point, what's the purpose. So remember, mitosis is for growth, repair, and asexual reproduction in bacteria that do that. But meiosis is specifically for making these kinds of cells and participating in sexual reproduction. So here are some key terms that are pretty important to kind of make clear before moving forward. So if you need to kind of for sure jot these down before moving on with the rest of the recording. I think it'll help you to have that reference, because when I use these terms, it's really helpful to have it jotted down so you can realize, wait, she keeps saying somatic cell. What does that mean again? Look down. Oh, okay. So somatic cells are kind of normal body cells. So any kind of cell that's not a reproductive cell. So a somatic cell could be like a skin cell, a liver cell, a hair follicle, a toe cell. You have a lot of different kind of specialized cells that do different things. But somatic cells are referring to anything that does not have to do with reproduction. So the word soma means body. So yeah, it's just like a normal body cell. Versus gametes. So gametes refer only to reproductive cells or sex cells like sperm or eggs. So somatic cells are basically anything else, and then gametes are just these one kind of cells. Reproductive cells, sperm or egg cells, and that's it. So when I refer to, somatic cells as anything else, gametes, only those. And so there is a specialized kind of cell that has to be able to divide to become gametes, and those are called germ cells. So it's not like your skin cell can make gametes. No. There are some specific cells that this is just their job, to become gametes and like divide into those guys. So germ cells, I don't know why they're called that. They don't have anything to do with germs. So we call germs, like bacteria and viruses and whatever. I don't know. It's probably one of those things. It's like when they discovered it, they were like, that's a germ, or I don't know, whatever. So I don't know why it's called germ cells, but those are the specialized cells that produce gametes, like eggs or sperm. So they have a very specific job, which is making these guys. And so when a gamete fertilizes another gamete, then that cell is called a zygote. So then that is one cell, and we call that a zygote, and it will end up dividing and then, becoming the new organism. So those are going to be the key terms. So as we move forward, when I say them, it might help to have those for reference. And so a couple more key terms. In biology, there's so much vocab and terms that, we make up for things, but it does end up being easier having a word for a scenario that you can say versus having to explain like, oh, it's that kind of cell that only has one copy of each chromosome versus the other one that has two copies of each chromosome. If we have a word for it, it's a lot easier to describe as we go. So, we have these two terms that describe how many copies of chromosomes a cell has. So if a cell is haploid, then it only has one copy of each chromosome for that species. So remember, there's different species that have different number of chromosomes, but we have this letter N that is kind of like the variable, the stand-in for how many chromosomes that species has. So for example, in humans, N is 23. So if a cell is haploid, then it has 23 chromosomes for that species, for humans. If a cell is diploid, di meaning 2, then the cell has 2 copies of each chromosome for that species. So you have one from mom and one from dad, and that makes 2 N. And then remember, N is 23 for humans. So if you get 23 from your mom and then 23 from your dad, you have a total of 46 chromosomes. So diploid, di meaning 2, you get 2 copies of each chromosome for that species. So now that makes sense, like that karyotype picture that I showed, I think it comes up again in a couple slides. That's how you have 2 copies of each chromosome for that species, because you've got one set from mom and one set from dad. So somatic cells, any normal kind of body cell has 2 copies of each chromosome for that species. They're diploid. And gametes are haploid. They only have 1 copy. And then germ cells are 2N, diploid. But because they're going to go through this whole process of meiosis and make gametes, they make haploid cells. So again, I think this terms, it might be a little confusing at first, but it just kind of takes practice, and it is faster to apply those terms instead of having to describe each time, So again, if we kind of like combine all these terms, somatic cells are normal body cells, and they are diploid. So they have 2 copies of each chromosome. Germ cells also are diploid, but because they're going to go through this process, and we'll show how, they will end up becoming or giving rise to gametes that are haploid. And then when the gametes fertilize, so if I say this is 1N and this one is 1N. When fertilization happens, that zygote is going to be 2N because they got one copy of each chromosome from dad and one from mom. And then, like I said, N is the number of chromosomes for that species. So in humans, N is 23. So gametes each have 23. This is kind of ugly, sorry. So this one has 23. And the total in a zygote and then in you is going to be 46 chromosomes because you got one set from mom and one set from dad of each chromosome. And that is what gives rise to that picture of all the different chromosomes that I showed in a karyotype, and to the homologous chromosomes. So each pair is called a homologous chromosome. They code for kind of the same things. They're the same recipe book, and then this is a different kind of recipe book, and this is a different kind of recipe book. And so you get some recipes from your mom and some from your dad. And so fertilization is how you get one of each. Yeah. So hopefully you're with me so far. So meiosis does have many of the same mechanisms as mitosis, but remember they're totally different purposes, and that's the big difference. So mitosis is just for making cells for growth, repair, and asexual reproduction, and it happens in somatic cells. So all your body cells, again, like your skin cells that you're constantly shedding and stuff, you need to make more, so you do that through mitosis. And it separates into just two identical daughter cells that are diploid. So those cells each have the full number of chromosomes, versus meiosis, which is different. And why is it different? Like, why can't we all just divide like that? It just happened through years and years, and like this weird development and whatever. So because sexual reproduction requires fertilization, two different cells coming together, if those two different cells had the full number of chromosomes and came together, then now that offspring has 4 times the chromosomes. And every time that organism, then divides and then ends up, having fertilization also, every time it's going to double and double, and that's just not sustainable. Like, cells just can't do that. They can't express that much information. So there has to be a way of like dividing the information up in half, and then when it recombines, it makes the full number. It just works out that way better than, yeah. So if the two cells each had one set of chromosomes, then the new cell has 2 sets of chromosomes. So yeah, for the reproductive cycle to continue, you have to kind of cut the number of chromosomes in half before fertilization, or else it would just be doubling and doubling and doubling every time. So that's why there is meiosis or meiosis. In case you're ever having like trouble, with the terms, like flipping which one is which. Mitosis, you can think my toe. If I stub my toe and I have a cut or something, like I need to make more cells through mitosis for that random toe. Versus meiosis. Meiosis is how I was born. It's how I made me, or whatever. So meiosis or meiosis, it happens only in specialized cells, the germ cells. So this guy would be starting. It's only used for making the specialized gametes in sexual reproduction. And then at the end, you actually end up with stuff that's different. So at the very end, you have 4 different cells, and they end up being genetically different from each other and from the parent. And, you know, we'll get into why. And each of them are haploid. So they're small details, but they make a big difference. So you make 4 different cells. They're genetically different, and they're haploid. So they only have 1 copy of each chromosome. So even though like these sizes kind of look similar, they are different. They should have made like a lot smaller or something. Yeah, so they have different purposes and they have different like outcomes. Yeah. So let's get into meiosis. How does this happen? Again, we're going to go into more detail, but remember the big picture. Why is it happening? For making gametes in sexual reproduction. So most animals and plants are diploid, having 2 sets of chromosomes in the somatic cells. And so in meiosis, the starting cell is diploid, but the ones that you make, the gametes, are haploid. So yeah, you're ending up with half the amount of chromosomes in a gamete. So to reduce the chromosome number, meiosis does duplicate the DNA, but because there's 2 rounds of division, that's how you end up with half by the end. So we'll see that, in the pictures moving forward. So the G1 phase, so interphase does happen before meiosis also. So a cell needs to prepare to divide twice. So it makes more proteins and stuff like that. It copies the DNA. So G1 of interphase before meiosis focused on cell growth. In the S phase, again, the chromosomes are copied or duplicated or replicated. There's different terms, but they're copied. And again, S stands for synthesis, so that's how you can remember. The S phase is when the DNA is synthesized. And then the G2 phase, growth 2, the cell undergoes the final preparations for meiosis. So again, then the small meiotic phase is meiosis and cytokinesis. And so this is basically the same kind of image, but for meiosis. So there are 2 rounds of duplication, so meiosis 1 and then meiosis 2. So we'll go through meiosis 1 first. And again, I like this image, this kind of image, because it kind of simplifies everything into just following chromosomes. Yeah. So before interphase, there is 2 chromosomes, right? 1 of each, because this cell is diploid, di meaning 2. So there's a dark blue and a light blue. And so after interphase, when the DNA is copied, now you have 2 light blues and 2 dark blues. During prophase 1, so there are technically the same phases as mitosis. It's just 2 rounds of division. So it goes through prophase 1 and the whole thing, and then prophase 2 and then the whole thing. So prophase 1, those chromosomes pair up. So these are sister chromatids, the light blue. The dark blue ones are sister chromatids. And then when we call them when we refer to them together, they're homologous chromosomes. So the blue chromosomes are kind of the same color, blue, just different versions. So they're homologous chromosomes. So in prophase, all the chromosomes pair up and, the cell prepares. Metaphase, they meet in the middle, but the main difference here is they meet up side by side. So the sister chromatids are side by side, and again, we refer to them as homologous chromosomes. But in metaphase 1, the homologous chromosomes get pulled apart. So metaphase, they meet in the middle, but next to each other. The meiotic spindle, so the fibers reach out, Spider-Man web, grab on, and then pull each apart. So the chromosomes are pulled apart, And meiosis is kind of crazy because at this point in time when they're next to each other in prophase and metaphase, they physically kind of switch some information. So if you can see my fingers, they physically are so close to each other they can kind of switch information. They like twist up and then like branch off and swap pieces. So then when they get pulled apart in anaphase, you can see now there's a mixture. So on one of the sister chromatids, there's dark blue and light blue. And on the other one, there's dark blue and light blue because they kind of swapped pieces from each other when they were next to each other. So in anaphase, when those chromosomes are pulled apart, they're not all identical to each other anymore. There's a blend on some of them. So anaphase, those homologous chromosomes are pulled apart. And then telophase and cytokinesis happens, but then each of these cells divides again. So now in the next slide, in meiosis 2, each of those 2 cells that you made is going to divide again. So prophase 2, you know, they pair up, they prepare. Metaphase, they meet across the middle. Anaphase, they get pulled apart. The sister chromatids get pulled apart. And then telophase and cytokinesis happens, and then, you know, the nucleus reforms, the cytoplasm is separate, and now at the very end you have 4 cells. This slide calls them granddaughter cells, but I think we just call them daughter cells. So at the very end we have 4 cells. They are haploid, so they only have 1 copy of each chromosome. And they are genetically different because of that process called crossing over. Now, this one is all dark blue, but this one is a mix of dark and light blue, and this one is a different mix of dark and light blue, and then this one is a mix of light blue. So, for example, sperm cells, like these, even though maybe they came from the same guy, they have such different combinations of genetic information that, you know, they would have a different looking zygote. So this is why even if you and your siblings come from the same parents, you guys don't look exactly the same because you are made from cells that have different combinations of genetic information. And then there's the random chance that this gamete will fertilize a different one, or this gamete will fertilize a different one. And then, yeah, on the other end, like, there's so many different possible combinations of genetic information that's why you don't look just like your parents and you don't look just like your siblings. You look very unique because you have a unique blend of genes that make you you. And again, remember, like, through this picture we only showed 1 chromosome, but we have 23 pairs of chromosomes. So if you think of this thing of crossing over happening with all the other chromosomes also in that cell, there's so many, like, endless combinations of genes and traits and stuff from either your mom or your dad, and then for sure, like, the random chance that one specific cell will fertilize another specific cell. Like, there's so many different combinations. So yeah, that's why you don't look different, or you don't look exactly the same as your siblings unless you're an identical twin. Yeah. So again, to compare, meiosis versus mitosis. Meiosis, different purposes. So meiosis used only for making gametes for sexual reproduction. And you make, at the end, 4 cells that are genetically different, and they're haploid. They only have 1 copy of each chromosome. And mitosis, even though it is kind of similar, like they're the same name for the phases, mitosis, remember, is a totally different purpose. It's just for asexual reproduction, growth, and development. And it only makes 2 cells that are genetically identical to each other and to the parent cell and their diploid. So yeah, I know it's a lot of information, but piece it together, like, you know, and with the terms. just like one piece at a time and fitting all the information together. But think of the big picture, basically. So I kind of already talked about this, but how does meiosis create so much variation? It's those two things of independent assortment, which is saying that, like, if we were to track more than one pair of chromosomes at a time, the way that they line up in the cell is totally random. So even though we can picture it like, oh, you know, 2 blue are going to line up at this side and 2 red are going to line up at this side, that's one possibility. But that's just kind of for ease of showing, you know, teaching it. But it's also possible that when they happen to meet in the middle at metaphase, that all the chromosomes possible in the cell can meet in a different way. So in this case, on the bottom, it could be that the bigger chromosomes, the blues on one side and the reds on the other side, and then the other chromosomes line up a different way. So it doesn't have to be like both blues go on this side, both reds go on this side. It could be like just a random alignment. So that's one way that it's like, whoa, there's so many different ways that this can go down because there's different chromosomes. There are more than one pair. They can all arrange in different ways. So that is a way that, like, that's a lot of genetic variation. So I don't know why this picture is blurry, but here's another one that's kind of showing the same thing. So possibility 1, if we follow now 2 sets of chromosomes, here's a bigger chromosome and then a smaller chromosome. It could be that the 2 blues line up on one side and the 2 reds line up on the other side. But it also could be possible that they line up on opposite ends. And so that's why at the end, if you track it, like, oh yeah, that leads to, at the end, different possible combinations. Versus on the left or the right side, like, those are different possible combinations. So it does make a difference how they line up, and it's totally random. So that is one way that there is a lot of, like, genetic variation. There's no telling, like, what combination of traits you're going to pass on. Especially with that thing that I called crossing over. It's also known as recombination, but it's the same thing. It's crossing over. So like I said, those 2 homologous chromosomes being so close to each other, they can physically switch information and, like, break off and then swap those pieces. And so that's why you get that mix of, in the pictures, like, part blue but part red, and then this one part red but part blue. So crossover happens between homologous chromosomes when they're close together and they exchange material, and so that's another way that there's even more variation, because even the 4 cells that are in this cell, they're not going to get all the same mixture of chromosomes. So yeah, that's another reason, like, yeah, you and your siblings, again, don't look exactly alike, because there's these 2 mechanisms working that makes so much variation. So it's independent assortment and crossing over. And so we kind of have discussed, you know, what happens when something goes wrong in mitosis. So remember, there's different purposes. So in mitosis, if something goes wrong and, like, DNA is altered or the wrong number of chromosomes gets, like, pulled to one side, it could lead to errors and uncontrolled cell growth, cancer. And so mitosis, remember, that's a different thing. But meiosis, what happens when something goes wrong there? Because you're influencing, like, another potential organism, it's kind of it's different. So what happens when something goes wrong in meiosis? That leads to inherited disorders, because these cells are specifically for reproduction. That's actually going to cause something wrong in, like, a separate individual, you know, a separate person, the zygote and the baby. So when something goes wrong in meiosis, they can lead to inherited disorders. So there are different kinds of chromosome disorders. So it can either be like the wrong chromosome number because maybe like they didn't pass on the right number of chromosomes to each other. Or it could be like structural rearrangements where like maybe one part of a chromosome like breaks off and now it's missing that information. So even small parts of chromosomes can have a lot of genes. So in a lot of cases, chromosomal disorders are dramatic and often fatal. So here again is a picture of a karyotype, so just a way to show, chromosomes. So the karyotype shows the number and appearance of chromosomes based on like the length, and then if we add dyes, and yeah, different structural things. So that kind of can help us see like what happened. If something goes wrong, if somebody has one too many chromosomes or one too few chromosomes, that's where you would see it by doing a karyotype. So a way that can happen is this situation called non-disjunction. So normally after meiosis, each gamete gets one copy of each chromosome. And non-disjunction is when some error happens and the right number are not passed on correctly to each side. So it can happen in either the first division or the second division. So here in this picture, if it happens during the first division and the two homologous chromosomes don't get separated, they both go to the same side, that is going to end up having some gametes have too much genetic information, one extra chromosome, and then on the other side some are going to be missing, so these gametes are going to be missing a chromosome. So yeah, ideally each one of these cells would have one big chromosome and one little chromosome, whether it's red or blue of each. There should only be one of each, one big and one little. And so here you can see there's two bigs and one little, and then here they're both missing a big chromosome. So yeah, if they don't get like divided up properly, some are going to end up with too many, some will end up with too few, and that can happen either in the first phase of meiosis 1 or in the second division in meiosis 2. So here, if it happens here, then that means these are going to have, this one's going to have an extra and this one's going to have one too few. So that's what non-disjunction is when the chromosomes aren't divided up correctly. And that leads to what we call aneuploidy. So that's when you have like either one too many or one too few of a chromosome. So human zygotes, again the fertilized cells that are missing one copy of a chromosome, usually don't develop to birth because they have only one copy of essential genes. So yeah, if you're missing a copy, then usually that zygote just won't develop. It will often end up in miscarriages because they don't have all the essential genes. If there's only one copy, that's just not enough. And in cases with extra chromosomes, they usually also fail to develop to birth because it's just too much information, and your cell's trying to code for all these different things, like way too much stuff is just not possible. So even if you end up with extra chromosomes, they're usually gonna fail to develop to birth. But if it happens to be a chromosome that's kind of small, so remember on the karyotypes there's like bigger ones and then there's little ones. If it happens to be an extra chromosome of the little ones, there might be more chance that that offspring will survive weeks to many years, because it's not like a bunch of extra genes like if it was a big chromosome. It's like a little bit of extra genes, but it is not really like feasible, so it does depend on what chromosome it is, what the kind of like survival rate would be. Yeah, so the smaller chromosomes, maybe, it can result in offspring that survive for several weeks to many years of the smaller chromosomes if there's extra. But yeah, you might be tempted to think like the more genes the better, right? Like it doesn't matter if you have extra chromosomes. No. Cells are made to and like calibrated to only make a certain amount of stuff. And so it's only anticipating two doses of each gene. So having a third one disrupts the balance. Like it's just not possible. Yeah, so it's not like the more genes the better. If you have a whole extra chromosome, it's not good. Sometimes, like I said, there can be either the wrong number of chromosomes or there could be structural changes to chromosomes. So here are a couple or a few of the structural changes that can be. So instead of just like a whole extra chromosome or missing one, sometimes something will happen where it's like a part is broken off of a chromosome, or it's broken but then put back in the wrong way. Yeah, so there can be what we call deletions, where part of a chromosome like goes missing. A duplication, where now there's 2 times as many as of like one part of a chromosome. Or it could be that a part got pulled out but then put back in the wrong direction, and so that's what we call an inversion. So all of these do they have an effect, but they're not as serious as having a whole extra chromosome or missing a whole chromosome. So like these could make a big difference, especially if some genes get deleted. It could make a difference that these genes are now double. And it could make a difference that these genes are now put back in the wrong way, because that alters like the recipe. Like say you were reading a recipe and all of a sudden it has the last steps first. That could still make a difference. But I would say having the wrong chromosome number is probably worse than like a structural rearrangement. So I know this was a lot of information, but hopefully, again, remember the big picture, like what is the point of meiosis? Let's go back to the learning objectives. So hopefully now it kind of makes more sense. So hopefully you'll be able to explain how the variation that's possible because of sexual reproduction is like an advantage evolutionarily. Hopefully you can kind of describe the behavior of chromosomes during meiosis. So, okay, there's homologous chromosomes, they get divided up, and then the sister chromatids get divided up. Describe the cellular events during meiosis, what is the difference between meiosis and mitosis, and then what are the mechanisms that meiosis makes so much variation possible. And so again, that's independent assortment and crossing over. So I know that was a lot, but try to think big picture.